Welcome to MycoTalks. We'll get started momentarily once people have joined. Welcome everyone to MycoTalks, hosted by the MRC Center for Medical Mycology at the University of Exeter. My name is Christina Cuomo, and I'm a professor at Brown University. And the co-chair today is Professor Jay Coles from Tulane University. Our speakers today are Linqi Wang from the Chinese Academy of Sciences in China and Professor Heather Conti from the University of Toledo in the United States. MycoTalks are organized by the committees shown here on the slide, with online assistance running the event from Jenny Spencer and MRC Center staff. MycoTalks are held at 4 p.m. UK time, usually on the last Thursday of every month, and we aim to promote diversity in our field regarding career stage, gender, geographical location, and research area. Previous talks are all available on YouTube in the MRC CMM channel, and the committee is always happy to receive speaker suggestions on our future program. The format consists of two 30-minute talks on different areas of medical mycology, followed by a 30-minute Q&A session at the end. To ask a question, please post it in the Q&A tab, which the moderators will see, and please specify the name of the speaker to which the question is addressed. The chairs will alternate questions between the speakers at the end and may choose to combine questions that cover similar topics. All questions posted in the Q&A will be sent to speakers after the event, so they will still receive them even if we run out of time to address them all. The upcoming MycoTalks are listed on the website, and you need to register for each session. The two final sessions of this fifth series of MycoTalks will be held on June 26th, when Andy Alspaugh and Erica Washington from Duke University will be presenting, and on July 24th, when Jamie Konopka from Stony Brook and Dane Usher from the University of Exeter will be presenting. You can also register to attend MycoClinics, an online series of talks specifically focused on clinical topics. And the upcoming talk is on June 5th, focused on aspergillosis. You can find schedules for MycoClinics on the MRC Center website. And the organizing committee for MycoClinics is listed at the bottom of this slide. It is now my pleasure to introduce our first speaker, Linqi Wang. I'll go back. Linqi earned his BS in microbiology from Shandong University and obtained his PhD from the Institute of Microbiology in the Chinese Academy of Sciences. He was conducting his postdoctoral work from Xiaorong Lin at Texas A&M. And then he joined the Institute of Microbiology in the Chinese Academy of Sciences as a professor. His team demonstrated that phenotypic resistance can arise during fungal infections and contribute to antifungal treatment failure. Additionally, he developed novel strategies targeting phenotypic resistance, which he showed outperformed existing treatments in fungal meningitis models. In a collaborative study, he identified an emerging invasive fungal pathogen and revealed that host body temperature-induced mutagenesis drives the emergence of pan-drug resistance and virulence traits in this pathogen. He's also identified evidence that microbial quorum sensing can trigger eukaryotic or fungus-specific behaviors, such as sexual reproduction and meiosis. He serves widely as an editor in many international journals, and is a F1000 faculty member. Welcome to MycoTalks, Linqi, and we are very looking forward to your presentation, and the stage is yours.
Thank you, Christina, for the nice introduction. Okay, let me share my slides. So can you see my slides?
Yes.
Okay, so nice. So, so I would like to thank the organizers to have me here. Okay, so I have this great opportunity to share our recent findings with you. It's about host-induced fungicide phenotypic resistance. My name is Linqi Wang. I'm from Institute of Microbiology, Chinese Academy of Sciences. And my English is not good. And if I cannot get your the point of your question, so please send me email to this address, and I will draft a formal response letter point by point, I promise. Okay. So I would like to start my talk with these slides. So as we know, fungal infection can cause more than 3.5 million deaths per year with very high mortality. Let me do this. Okay. So so the fungal infection can cause more than 3.5 million deaths per year with very high mortalities, usually above 20%. In addition, I would like to say, so these slides can also tell us another important piece of information. So we can see most of these fungal pathogens are marked in red. Okay, so this means they are not obligate pathogens and not commensal pathogens. They are of environmental origin and called environmental fungal pathogens, which is the major group of human fungal pathogen. Okay. So in our lab focus on the central question regarding environmental human fungal pathogen, that is why this environmental fungal species can cause fatal infections and be difficult to treat. So as I mentioned at the beginning, a major clinical problem with fungal infections is they are usually associated with very high mortality rates. So one reason for this is we only have limited options to treat fungal diseases. Another reason is treatment failure. So actually happens very frequently. Okay. So let me take Cryptococcus meningitis as example, which is one of the most dangerous fungal diseases. And this disease is mainly caused by environmental human fungal pathogen Cryptococcus neoformans. And amphotericin B is the only available fungicidal drug to treat this disease, and the other drugs are fungistatic drugs. Unlike fungicidal drugs, fungistatic drugs can only inhibit the growth of Cryptococcus cells but cannot kill them. So if amphotericin B-based therapy fails, the mortality rates can reach as high as 70%. That is very dangerous. Okay. So in addition to Cryptococcus meningitis, amphotericin B is the last line of defense in many different fungal diseases because of its remarkable fungicidal activity. So actually, another advantage of amphotericin B is its resistant mutants are very rare clinically. A previous study from Susan Linqui's lab found that amphotericin B resistance mutations can confer a high fitness cost, and the resistant mutants cannot enable a successful infection in a mouse model. And since amphotericin B resistant mutants are rare clinically, so this cannot explain why amphotericin B based therapy fails in many cases. So as we know, amphotericin B is thought to be the final barrier for many different fungal diseases, as I mentioned before. Okay, understanding this question so may be very important. Okay, so for many bacterial and microbial parasite, a specific form of resistance called phenotypic resistance is an important contributor to antimicrobial treatment failure. So what is phenotypic resistance? So phenotypic resistance refers to phenomenon in which genetically susceptible pathogen cells survive high doses of cyto drugs without a change in MIC. And since phenotypic resistance involves in cyto drugs, so it works different from fungistatic azole tolerance, which has been reported by Professor Judy Berman's lab. And azole tolerance, as we know, azole tolerance is very important because Judy's lab demonstrated azole tolerance is tightly correlated with clinical outcome. But in comparison, the importance of fungicide phenotypic resistance remain to be established. So because there is no direct evidence supporting, so phenotypic resistance can be directly induced by host-derived factor in vivo during fungal infection. So what is the bottleneck? So in this field, so we believe the bottleneck is how we discover host factor stimulating fungal phenotypic resistance, because we have so many different host factors in our bodies, right? So to this end, we have developed a systematic approach which allows a comprehensive evaluation of triple interaction between host metabolites, amphotericin B, and the cryptococcal neoformans. And by means of this approach, we identified some metabolites can strongly induce tolerance of cryptococcal neoformans to amphotericin B. And so we are very, actually we noticed that, so among these metabolites, glucose has the strongest effect. And actually we are very excited about this finding, because as we know, our brains is rich in glucose. And the concentration of glucose in our brain can reach as high as millimolar. So that is a very high level, okay? And cryptococcal neoformans happens to be the leading cause of fungal meningitis. And in USA and in China, more than 70% of fungal meningitis cases are caused by cryptococcal neoformans, okay? So next, we want to know whether or not glucose in our brain is sufficient to trigger phenotypic resistance in cryptococcus neoformans. To test this idea, and we collected samples of CSF from patients with cryptococcus infections or other diseases. And we also use an enzyme called GOX, glucose oxidase, GOX. And this enzyme can catalyze the conversion of glucose into gluconic acid, okay? And based on our systematic approach mentioned before, unlike glucose, glucosic acid does not have an effect on the induction of phenotypic resistance in cryptococcal neoformans. So this enzyme-based approach allows us directly test whether or not brain in CSF samples is sufficient to trigger phenotypic resistance in cryptococcal neoformans. Okay, so as shown here, and if you use GOX to remove glucose from CSF samples, okay, you can see the levels of phenotypic resistance decrease dramatically. And if you add the sugar back, and you can find, okay, the phenotype comes back. So this means glucose is the key factor driving phenotypic resistance in human CSF. Okay. So next we want to know, so what is the key factor mediating glucose-induced phenotypic resistance? As a result, we identified transcription factor MYC1. So actually, this is very easy so for us to figure it out, because so MYC1 in different fungal species, MYC1 plays a very conservative role in sensing glucose. And if you knock out the MYC1 gene, so in cryptococcal cells, actually, so you can find the glucose-induced phenotypic resistance almost disappear. So that means MYC1 is the key transcription factor mediating glucose-induced phenotypic resistance in cryptococcal neoformans. Okay, next we ask the most important question so in this project. That is, okay, whether or not a brain glucose-induced phenotypic resistance can eventually compromise the efficacy of amphotericin B against cryptococcus meningitis. And if the answer is not, so that means this phenotypic resistance may not have a clinical significance. We don't have to bother with that, okay? So to test this idea, we used the mouse model of fungal meningitis infected with a viral type of MYC1 mutant cells. And we found without amphotericin B treatment, there was no difference, okay, in terms of survival curve of mice. This suggests MYC1 is not important for a brain infection caused by cryptococcal neoformans. But in contrast, so when you treated mice with amphotericin B for 2 weeks, then you stopped the treatment, you can see the big difference, okay? So in mice infected with viral type cells, all mice died within 3 weeks after stopping treatment, so because of relapse. okay? But in mice infected with mutant cells, MYC1 mutant cells that have lost the ability to induce phenotypic resistance, and 60% of mice are eventually cured. So cured means you cannot detect any living cell in mice brains. So these results indicate MYC1 mediated phenotypic resistance can eventually affect the therapeutic efficacy of amphotericin B against Cryptococcus meningitis. So next, we want to know the mechanism underlying MYC1 mediated phenotypic resistance. Okay, so as we know, so amphotericin B kill fungal cells through binding to the membrane component ergosterol, right? And we found in the presence of glucose, and MYC1 can regulate the production of different membrane components, especially the membrane component IPC, okay? So, and furthermore, we performed SPR analysis, okay? So this data, the data that can tell us two pieces of information. One is IPC can directly bind ergosterol with affinity even higher than amphotericin B. Another information is the presence of IPC can limit the binding of amphotericin B to ergosterol. Okay, so let me summarize the mechanism for brain glucose-induced phenotypic resistance in Cryptococcal neoformans. And we found in the presence of brain glucose, and the transcription factor MYC1 can promote the production of a membrane component IPC. And IPC can use a tag-of-war mechanism. And IPC can directly compete with amphotericin B for its target ergosterol, which makes MYC1 mediated phenotypic resistance happen. Okay, so since we already know IPC production is critical for glucose-induced phenotypic resistance, and we hypothesize inhibition of IPC production may enhance the efficacy of amphotericin B against the Cryptococcus meningitis. So we focus on the inhibitor for IPC synthesis, that is aureobasidiate, ABA. And as we know, ABA has very nice safety because its target AOR1 does not have any homolog in human genomes. So it is the fungus specific. But this drug was eventually abandoned by a Merck company because it has a narrow fungicidal spectrum. And our mouse model showed ABA is not useful for treating Cryptococcus meningitis. But when you use the ABA amphotericin B combination, you can see a very, very promising efficacy against Cryptococcus meningitis. So as I mentioned before, so Cryptococcus meningitis is one of the most dangerous fungal disease. Okay, so if you use the animal model, 7 days of treatments cannot cause a cure with any clinically recommended strategy. But if you use the ABA amphotericin B combination to treat the mice, the cure rate can be as high as 70%. So, and this number is promising. Okay, so in addition to the brain, our lungs are another organ commonly infected by fungal pathogens, including Cryptococcal neoformans. But unlike glucose, our brain, so our lungs are the glucose poor organ. Okay, and our in vitro drug killing assays showed the phenotypic resistance cannot be induced by glucose at lung-like concentrations. But previous study demonstrated fungal cells during lung infections are also difficult to clear with amphotericin B. So this may suggest phenotypic resistance may also occur during lung infection, but it uses a mechanism different from the one they identified in brain. And my postdoc, Dr. Kevei noticed the 2 papers from Dr. Drummer and Dr. Alino lab from Pasteur Institute. And they found the Cryptococcus can form dominant cells during lung infection. So as we know, in bacterial, the dominant cells can mediate a specific form of phenotypic resistance called drug persistence. Okay, so we hypothesize, okay, maybe the dominant Cryptococcus cells can also mediate the drug persistence. Okay, so during lung infection. So So next, we want to know whether or not cryptococcal neoformans can form amphotericin B tolerant persisters in vitro or during lung infections. Okay, we test this idea according to the definitions of drug persistence. So first, the cells don't undergo proliferation because they are dormant. So for cryptococcal neoformans, it's very easy to collect the dormant cells, and we only need to grow cryptococcal cells to stationary phase. And as shown here, okay, stationary phase cells look like bees without budding, but for growing cells, okay, so you can clearly see budding there. So this means they are proliferating. Okay. Importantly, the survival rate of dominant cells is 100 to 1,000 times higher than that of growing cells, with less than 1% of cell population surviving. So our results are consistent with the definitions of drug persistence, suggesting the cryptococcal neoformans can form amphotericin B tolerant persisters in vitro. Okay. Next, we want to know whether or not such a fungicide persistence occurs during cryptococcus lung infection. And to do this, we use an omic approach to identify a cryptococcus protein named SPS1. Okay, so okay, the expression of SPS1 is dormancy specific and is one of the most abundant proteins in dominant cells. So the gene encoding SPS1 can be used as a reporter gene to indicate the cryptococcus dormancy. And in infected mice through inhalation, so with cryptococcus cells carrying, okay, so the SPS1 reporter system. And we found that 7 days after an infection, we can clearly see a population of fungal cells with a high expression of a dormancy-specific protein SPS1 in the lungs here, okay? And we sorted this population of cells using flow cytometry and found that they were highly tolerant to amphotericin B. Furthermore, we infected mice again, so with SPS1 expressing dominant cells sorted from mice lungs, as shown here. And we found SPS1 expressing cells has intact ability to cause disease. Okay. So we proved the cryptococcus neoformans can form fungicide tolerant persisters during lung infection. So next, okay, let me see. So next, we want to know the determinant underlying the formation of amphotericin B persisters. So we found dominant cells has similar aldosterol level to growing cells, but have a higher RS resistance. And this suggests there may exist an antioxidant system induced in dominant cells, which may be responsible for amphotericin B persistence. So we took advantage of a metabolic profiling approach and identify the antioxidant aldosterone. Okay. We found the dominant cells accumulates high levels of aldosterone, but it cannot be detected, okay, so in growing cells. And if you knock out its biosynthetic gene, the EGT1, okay, and you cannot detect aldosterone production even in dominant cells. So the EGT1 is the important biosynthetic gene, so for aldosterone production. Importantly, we found intracellular aldosterone promotes the formation of amphotericin persisters in vivo and in vitro, because when you blocked aldosterone production by deleting the EGT1 gene, so and you will find so deleting EGT1 gene dramatically reduced the levels of amphotericin B persistence in vivo and in vitro. So furthermore, we found the effect of aldosterone on amphotericin persistence is conserved in different fungal species, including different cryptococcus species, including the cryptococcus gattii and Aspergillus fumigatus. Okay. So as I mentioned before, we identified ways to evaluate the formation of amphotericin B tolerant persisters in vivo and in vitro. So this allows us to explore the drug with promising efficacy against amphotericin B persisters. And in this regard, we use a drug repurposing approach, and we identify the antidepressant sertraline that are strongly effective in clearing amphotericin B persisters in mice lungs. So we believe that certainly may be a promising approach to compromise amphotericin persisters, at least in cryptococcal neoformans. So the last part of my talk is about another issue associated with environmental human fungal pathogens. That is, in the last 2 decades, so we can see many new pathogenic fungi have emerged, and most of them are of environmental origin. So our question is, so what role do environmental factors play in the emergence of pathogenic traits in environmental fungi? So in collaboration with China Hospital Invasive Fungal Disease Surveillance Network, and we performed taxonomic evaluation of more than 27,000 isolates from 96 hospitals. And as a result, we identified a previously undescribed invasive fungal pathogen. Okay, so I'm sorry, although I already published the paper and I still cannot make correct pronunciation for this new pathogen because it's have too long name. So I just use RF to refer to this new pathogen. Okay, so and we found RF is a multi-drug resistance pathogen. And this pathogen is resistant to all azole drugs and all echinocandins drugs. Unfortunately, 2 patients infected with this new pathogen died eventually, probably because of treatment failure. Again, so we use a drug repurposing approach and identify the bacterial antibiotic polymyxin B, which is strongly effective in killing this multi-drug resistant pathogen. Okay. So in addition, we found, so when you raise temperature, you found raising temperature can strongly promote mutagenesis, and which enables genomic evolution and the emergence of hypervirulence and pan-resistance, the variants. The pan-resistant variants means so the variants are resistant to all first-line antifungals, including all azole drugs, all amphotericin B, and all what is the last drug? I forgot that. Okay, echinocandins. Okay, echinocandin drugs. So okay, so this new pathogen is dangerous, and our results support the possibility that global warming may play a role in promoting the evolution of this new fungal pathogen. Okay. So, and our research has also triggered a public interest about global warming driving fungal evolution and emerging fungal diseases. But we don't have the answer for this big question, okay, since we only have one single evidence. And we are now testing approximately 800 fungal species for the effect of increasing temperature on mutagenesis rates. Hopefully, in the future, we will provide more data to support this important hypothesis. Okay, so finally, I would like to thank my collaborators listed here. Without them, I may not be invited to give a talk. And thanks to my daughter, and she drew the portraits of all the lab members. Okay, thank you very much. And I would like to take any question.
Thank you, Dr. Wang. Yeah, so we'll take questions after both the speakers. So, and just remember for people on the Zoom to enter your questions in the Q&A tab.
Okay.
This is Jay Coles from Tulane Medicine. My pleasure to introduce Dr. Heather Conti, our second speaker for today. Dr. Conti got her PhD at the University of Buffalo, where she made seminal observations on the role of IL-17 and IL-17 receptor signaling in a murine model of oropharyngeal candidiasis. She did a postdoc at University of Pittsburgh and now is associate professor at University of Toledo, and where she's gone on to continue to work on understanding host defense against Candida and the oral mucosa. And today, Dr. Conti is going to talk about the role of oral megakaryocytes and platelets and how that interacts with IL-17 signaling and for host defense in the oral mucosa. So welcome, Dr. Conti.
Thanks, Jay. I appreciate it. Let me share my slides. All right, everyone can see that? Awesome. All right. Thank you first to the organizers for giving me this opportunity. It's really a great honor to be here and to be able to share some of our recent work trying to understand further what the interleukin-17 or IL-17 receptor is doing in the oral cavity as it relates to signaling through oral megakaryocytes, and then how platelets are involved in protection against oropharyngeal candidiasis. So I'm of course preaching to the choir a little bit, and our first speaker already outlined this, but I think it's always worth mentioning that worldwide fungal infections are on the rise. The World Health Organization has listed Candida and Candida albicans in particular as a priority pathogen to guide research, development, and public health action. We even have the CDC with their Fungal Disease Awareness Week, and thus far it is still scheduled for September 2025. And all of this might be meaningless if the funding bodies don't support fungal research, right? And our colleagues and friends have really nicely illustrated this in a recent commentary. And I think that's an important thing to note. So Candida albicans is not just the benign commensal of the human mucosa and skin that we think it is, right? It can become pathogenic when the immune system is compromised, such as in persons living with HIV and cancer patients receiving radiation and chemotherapy. Candida albicans is the main cause of candidiasis, which can present as very different diseases. We have oropharyngeal, or OPC, as I'll refer to it, through to disseminated, which is the fourth most common nosocomial infection with a relatively high mortality rate of 40%, depending on the population. A really simple observation that can be made is that HIV positive patients are particularly susceptible to OPC, not to other forms of candidiasis, which means that CD4 positive T cells must play an important role in protection against OPC. So I'm going to assume everyone is at least marginally familiar with CD4 positive T helper cells. So I will just focus on the Th17 subset that differentiates in the presence of different cytokines like IL-6 and IL-1 beta. And of particular importance is IL-23, which is required for the expansion and function of this subset. Oops, I better get my pointer going. Th17 cells then go on to produce cytokines such as IL-17A, F, and 22 that are protective against extracellular bacteria and fungi, but pathogenic in some autoimmune conditions such as psoriasis. So IL-17A, which I'll refer to as IL-17, and IL-17F are part of the IL-17 cytokine family. And if we focus just on IL-17 and IL-17F, they exist as homo and heterodimers. They signal through the IL-17 receptor consisting of the IL-17RA and RC subunits. And we know quite a bit about the signaling mechanisms downstream of the receptor, a lot of that from Sarah Gaffin, whom I trained with. And I will not pretend that I had anything to do with determining the key adapter molecules downstream of the receptor, like ACT1, the TRAFs, or any of the molecules involved in RNA stability and translation, or the transcription factors like NF-kappa B, the CBPs, I-kappa B zeta, that are involved in new gene transcription. But what I was involved with was trying to understand all of these immune components in fungal infection. And we did this through the use of an experimental mouse model of OPC, originally developed by Scott Filler, where mice are exposed to Candida on day 0. And we do this by taking a pre-weighed cotton ball, applying the Candida suspension to that, and applying that sublingually in the mouse, right underneath the tongue, to an anesthetized mouse. Remove it, mouse goes on its way, and then throughout the time course, we take out the tongue tissue, we homogenize that, we plate that on YPD agar plates to get the CFU per gram of tongue tissue, which is our fungal susceptibility readout. And Wild-type immunocompetent mice, just like healthy humans, are going to clear the infection by day 3, 5 at the latest. And when we have mice deficient in either IL-23 or the subunits of the IL-17 receptor, these mice have a high fungal burden, high fungal susceptibility, meaning that these cytokines are critical in protection against OPC. And this was our first step in understanding overall the immune response to OPC, from recognition of Candida through to fungal clearance mediated by recruitment of neutrophils and different antimicrobial peptides like the defensins. And first, let's recognize that there are some missing pieces from here, right? This predates the recognition of candidolysin, which is a peptide toxin produced by the virulent hyphal form of Candida albicans that is important in orchestrating the acute response to Candida. Some cytokines are missing, IL-36, the IL-22 receptor, and its importance in different portions of the oral mucosa. But nonetheless, we narrowed down the CD4 positive T helper subset that is critical in protection against OPC. And I just want to take this opportunity, because we're all here, to just give the fungal research community a shout out and how absolutely supportive everyone is. This paper came out a long time ago. It was more recently named a JEM legacy paper. Very graciously, Mihalis and Lucas wrote a Pillars of Immunology article about it. So I just really want to thank everyone for that. And I promise you that this is not just going to be a lot of shameless self-promotion or reliving the glory days with the Gaffin Lab, but we did also narrow down in acute OPC, so the first time perhaps a baby is exposed to Candida, as opposed to an adaptive response, and the innate type 17 cells that produce IL-17. And I mention this because what this required were some experimental protocols that allowed us to probe live immune cells through flow cytometry. And we've continued with my group and really refined these protocols to be able to detect these rare immune populations like the oral megakaryocytes. And then my group has also focused further on trying to understand the role of the interleukin-17 receptor in the oral mucosa, the genes that are involved downstream of that, and how that leads to a protective response. And this was in this paper where we focused on the role of the interleukin-17 receptor in the oral epithelial cells specifically. And this is an important question to ask. Host defense against OPC is really IL-23 and IL-17 dependent, but you can ask, what is the tissue-specific role of interleukin-17 receptor? Because overall host defense in the oral cavity has contributions from many different tissue compartments: the oral mucosa, the salivary gland, the hematopoietic compartment through infiltrating immune cells and action in the draining lymph node. And it bears mentioning that the IL-17 receptor is expressed by both hematopoietic and non-hematopoietic cells. And at this point in that paper, we had eliminated the role of IL-17 receptor expression in hematopoietic cells. And we did this through a series of bone marrow chimera experiments. And I want to bookmark it. There is a point to this. We're going to get back to it. And when we talk about oral megakaryocytes, this will become important. So I consider myself an oral mucosal immunologist. And when I started my lab, I knew I still wanted to focus on oral candidiasis. But I became interested in how different forms of immunosuppression may affect the oral cavity, the oral epithelial cells, the IL-17 signaling pathway, the type 17 cells, the cells producing IL-17, the neutrophils, the antimicrobial peptides. And the first thing we did was focus on the damage radiation is causing to the oral mucosa and how that affects IL-17 signaling. But of course, a patient is receiving radiation or chemotherapy because they have a malignancy. And this is important to understand the connection between fungal infection and the incidence of cancer, and then how the radiation treatment affects this tripartite relationship. But as important as this is, and if anyone wants to discuss this at a later time with me, I would love to talk about some of the other projects in our lab. But today I will focus on thrombocytopenia as a form of immunosuppression or platelet insufficiency. And for some of us, that might seem like a strange thing to do, to discuss platelets as it relates to host defense. Makes sense, vascular diseases, but host defense, and in particular, antifungal immune responses. And I just want to say that that was the case for me as well, and definitely learned my lesson. This hopefully is familiar to a lot of you. This is actually from the textbook I use to teach the undergraduate class. And 10 years ago, when I started teaching this course, I did this. I just crossed off megakaryocytes and platelets. And I told my students, you have enough to worry about. Don't even worry about these cells. They're not involved in the immune response. And of course, they are the cells that I continue to study to this day. And hopefully this group is better informed than I was at that point. But if you do think of platelets in their role in hemostasis, I really can't blame you, right? They are anucleate cells derived from megakaryocytes that are absolutely critical for vascular integrity. But what we find is that they are dynamic immune cells. They release granular contents and microparticles, alpha granules, dense granules. They express on their surface and secrete different immune mediators, which allow them to have a really nuanced response during different inflammatory responses, and they can interact directly with other immune cells as well. So they have an important role in various vascular diseases and infections, including fungal infections. First, I'm not going to pretend that we were the first to connect platelets with Candida albicans, but nonetheless, invasive fungal infections are associated with thrombocytopenia, those caused by Aspergillus and Candida species. And these fungal infections are predictors of ITP. During fungal sepsis, platelets release immunologically relevant microparticles and releasates. And we and others have shown that platelets have direct bactericidal and fungicidal activity. And during aspergillosis, platelets recruit neutrophils and are important for protection. And then also, we do see that platelets respond to canallysin, that peptide toxin that I mentioned previously. And this is important to enhance Th2 and Th17 responses during allergy responses. So if we narrowly confine our understanding of platelets to hemostasis, I think we're guilty of that even more for megakaryocytes. And in no way is the biological relevance of megakaryocytes confined to their role in platelet biogenesis. What we're finding more and more is that, well, of course, there are platelet-producing megakaryocytes. There are these more immune-like megakaryocytes. And while we're still, others are still trying to understand the alternative developmental pathways that these more immune-like megakaryocytes might follow, they do seem to arise during times of stress, such as radiation or infection. And these what we can call bona fide immune effector megakaryocytes, they secrete cytokines and chemokines, they express various immunoreceptors, they're able to interact with other immune cells and can even present antigen to enhance Th17 responses. And while megakaryocytes are primarily found in the bone marrow, there is a distinct population in the lungs, and they tend to be these immune megakaryocytes. So I think, right, also at first pass, it makes sense that platelets would be involved in disseminated candidiasis, right, a bloodborne infection. So to make a leap into mucosal candidiasis, we must have had some hints along the way. And one came from a paper I had mentioned before, where we looked at the transcriptional profile in our infected mouse tongue compared to human oral epithelial cells. And this is just some pathway analysis, but what jumped out from that was this one gene, which encodes for p-selectin glycoprotein ligand 1, which binds to p-selectin on activated platelets. So when we looked at wild type versus our IL-17 RA knockout mice on day 1, we did see some differential expression of some platelet-related genes. Not terribly compelling. Platelet levels were lower in our IL-17 receptor knockout mice. Again, not thrombocytopenic, but they did have lower levels. And I think just in general, what has held back our understanding of platelets in host defense has been a way to knock them out, right? Deplete them, get rid of them in a mouse. We can use antibody depletion, which has some off-target effects. We can use a TPO or thrombopoietin knockout mouse, which is not very efficient in getting rid of platelets completely. And our collaborators accomplished this by developing a platelet depletion model where we take a PF4 Cree mouse, PF4 expressed by megakaryocytes and by extension platelets, and cross this to an inducible DTR mouse. And then upon diphtheria toxin administration, megakaryocytes and platelets are ablated. So we take this model and we add that to our OPC model. So mice received the DT injections every other day, starting in day -7. We infect as we normally would. And then we look at fungal susceptibility at different time points. We take out tissue to do flow cytometry, etc. And what we found, admittedly, quite surprisingly, I think, was that the mice deficient in platelets were highly susceptible to OPC, showing a high fungal burden here compared to a wild type mouse. And then and you remember, neutrophils are key for protection against OPC in the oral cavity. So if we look at these mice, why do they have such a high fungal burden? We see that in the mice, the depleted mice, there is a lack of a neutrophil influx into the tissue. This is doing flow cytometry on bulk tissue. And even when we look in the superficial layer where the infection is happening, we see a decrease in the number of neutrophils that are being recruited to the tissue. And then when we take our depleted mice and we adoptively transfer platelets and we add them back, we see that the neutrophil response is restored. And this goes along with a decrease in the fungal burden as well. We saw lower levels of some of the chemokines and growth factors involved in the neutrophil response during OPC. It doesn't necessarily mean that the platelets were producing these factors. And I think an even more interesting question we asked is, were there direct interactions between platelets and neutrophils in the oral cavity? So platelets are known to recruit neutrophils through interaction of p-selectin with PSGL1, and this is actually how platelets recruit neutrophils during thrombosis. And when we disrupted this interaction, we saw a decrease in the number of neutrophils recruited to the oral cavity, shown here, and showing that it was a direct interaction between those platelets and the neutrophils, getting those neutrophils into the oral cavity. So at this point, right, you should also be thinking back to that animal model and the fact that when we use that, the PF4 DTR mouse, we are not only right? We're depleting platelets, but we're doing that through depletion, through ablation of megakaryocytes. So you could ask the question, knowing that megs and platelets are both really important immune cells, are megs themselves playing a separate role from platelets in OPC? And taking that a step further, really supported by the finding of lung megakaryocytes, could there be a population of oral megakaryocytes? And we also have some success in finding rare immune cell populations. So we decided to give it a go and see if we could find oral megakaryocytes. And indeed, in wild type mice, so just sham mice, not infected, we see a small but consistent population of CD41 positive cells from perfused mice. And then when we infect these mice, that population expands. And that same expansion is not seen in the bone marrow. And then when we look for these cells, they are in the areas of infection. They're in the basal stem cell layer. They are not associated with red blood cells or blood vessels. And we see that they are gone in those depleted mice. And then in the interest of time, right, some of the questions we asked, right, these are not platelets. Platelets are small, megakaryocytes are big, so we detect them on different scales. So they're not platelets. They could be platelet aggregates. We are very careful to always use a nuclear marker. Platelets are anucleate, so we can tell them apart that way. And we've used a whole panel of different MEG markers. They are CD61 positive, which is the companion molecule to CD41 of the fibrinogen receptor. They are p-selectin positive. They're CD42D low, which is a receptor of the von Willebrand factor receptor. And they are proliferating in the oral tissue. Right? We think they are resident, or if you're comfortable, at least localized to the oral tissue. And importantly, they're not platelets coating neutrophils or macrophages. So they're CD11B negative, Ly6G, whatever, GR1, whatever marker you want to use for your neutrophils, F480 negative. And now what we are doing is platelets are obviously interacting with the neutrophils, and we're trying to understand the differences in the platelets that are coating some of these cells. Right, but our megs are not platelets coating them. And then also, all right, the number of megakaryocytes makes sense. They're not any more than what you would find in the bone marrow. I think if there were a lot more, we would be a little bit worried. They are on the same line, about the same number of macrophages that we find. The difference is that megakaryocytes expand and the macrophages don't during OPC. And I think, just like I mentioned before, right, another lesson learned is listen to your trainees. Dylan, the first author on our publication, really did a fantastic job refining the methods to be able to find this rare cell population. And these are all of the things I asked him to do. Every time he would come to me, he said, I really think there could be oral megs there. I was like, nah, they're just platelets. No, they're just aggregates. And he would have to come back and prove to me, show to me that they really seem to be oral megakaryocytes in the oral cavity. And as we started to immunophenotype these cells a little bit more, like lung megakaryocytes, but unlike bone marrow megakaryocytes, the tongue megakaryocytes express higher levels of CCR7. So we think that they at least have the capability of trafficking to the draining lymph node. They have increased expression of MHC, CD86, co-stimulatory molecule B7. And what's interesting about these more immune-like megakaryocytes in the lung is that they maintain some of their hematopoietic progenitor properties. And they're even able in mice to repopulate the bone marrow. And as part of this, they maintain their CD34 expression. A portion of our cells in the tongue also continue to express CD34, but it shows that there seems to be a diverse population within the tongue as well. So then we became interested in what some of the fungal signals are that might be controlling megakaryocytes. And we had some evidence in a cell line that direct contact between megakaryocytes and Candida was not necessary, at least for some functional outputs. So the perfect candidate to look at was Candida lysine. And this is also supported by work from Dave Corey's group that showed that platelets through Candida lysine, or I should say, excuse me, Candida through Candida lysine do activate platelets through interaction with GP1B alpha. And we decided to first go to our MEG cell line to understand how Candida was interacting with MEGs. And we see that indeed, Candida lysine is necessary for platelet production to elicit platelets from megakaryocytes. We see a lower level of platelet release when we use this EC1 or Candida lysine mutant compared to the revertant strain. And then when we collect those platelets and use them in a subsequent Candida killing assay, we see that only platelets from the revertant strain kill Candida after. And then moving that into our mouse model, we see the same thing. When we infect with that mutant, we don't see that expansion of the oral megakaryocytes like we do with SC5E8 or the revertant strain. And then for the Candida aficionados out there, when we use the commensal-like strain 529L, which expresses lower levels of Candida lysine, we see something similar. So then when we wanted to understand from the host aspect and what some of the cytokine signals that are necessary for this MEG expansion, we of course looked to IL-17, right? Because I'm an IL-17 person, but also supported by others who have shown that IL-17 is involved in megakaryopoiesis. And even when we look in the bone marrow of our IL-17 RA mouse, a sham mouse, we do see lower levels of bone marrow megakaryocytes. And then when we looked in the oral cavity, we do see a lower level of those cells in our sham mice, and we don't see that same expansion when IL-17 signaling is absent. Of course, this doesn't show is it direct or indirect? Is it through expression of the IL-17 receptor by the megakaryocytes? I've done some work in that cell line, but this might not make sense with some of our previous data. Remember, I had said that IL-17, we had eliminated the contribution of IL-17 receptor on hematopoietic cells through those bone marrow chimeras. And IL-17 signaling dogma really is that the effect of the receptor is through non-hematopoietic cells, mesenchymal and epithelial cells. And we had based this also on, like I mentioned, those bone marrow chimera experiments where we showed that irradiated IL-17 RA knockout mice that received wild-type bone marrow still had high fungal burden. And likewise, an irradiated wild-type mouse that received IL-17 RA bone marrow still cleared infection. This was shown later on with the non-canonical NF-kappa B transcription factor I-kappa B zeta that we showed with Sarah. Same thing, right? There didn't seem to be a role for signaling in a non-hematopoietic cell, or excuse me, a hematopoietic cell. Right, so what could explain that? Well, first, what we see is that megakaryocytes are the most radio-resistant myeloid cell. So bone marrow chimeras that are meant to eliminate these cells would not account for them. And when we look in the oral cavity at our oral megs, they too are radio-resistant. Right? They're maintained after radiation. And even if we went back and we looked at when we focused on IL-17 receptor signaling in the oral epithelial cells through our K13 Cree mouse, if we look at that conditional knockout mouse and compare that to a full knockout, yeah, I think there was some room for expression by other cells, right? Either in the oral mucosa or with our oral megs in that susceptibility, especially at the early time points where we see oral megs are involved in the response and platelets. So it would be, I think, paradigm shifting to really understand signaling in a hematopoietic cell in these oral megakaryocytes, in these in megs just in general, since very little is known about the immune role of megs, the role of interleukin 17. And in order to do these deep mechanistic studies that we've done in stromal and epithelial cells, right, we need a cell line to do that. And we have this human mega-1 cell line that we're able to look at its hemostatic properties controlled by IL-17, its immune properties controlled by IL-17. And we see these cells, of course, express the receptor. They respond to the cytokine like they do to other vascular agonists like TPO. They express antifungal immune mediators. And we're also able to take this system into our mouse model and take our PF4 Cree mouse and cross it to an IL-17 receptor flox mouse to start off with, or the NF-kappa B zeta, which encodes for I-kappa B zeta. And we can understand the signaling specifically in these hematopoietic cells during OPC. All right, so what do we think oral megs are good for? We think a lot in the oral cavity. They respond to Candida lysine, they expand, they produce platelets. These platelets interact with neutrophils. They produce different cytokines and chemokines, presumably. And that leads to protective recruitment of protective response through those neutrophils. These cells do seem to have a more hematopoietic progenitor-like phenotype. They express CD34. I think it's going to be interesting to try and understand the interaction between these cells and the oral stem cell niche. So there's obviously a lot to do trying to understand the origin of these cells. We are working now on the development, the megakaryopoiesis that is happening in the oral mucosa, what the intermediates are doing. I didn't go into it really at all, but as a megakaryocyte is developing, the intermediate stages have their own function, and we see even their own function in immune responses. So we're trying to understand which of those intermediates are involved during OPC. And then, like I mentioned, we're trying to understand kind of the crosstalk between these progenitor cells and perhaps with the oral stem cell niche, the vascular, the secondary lymphoid compartments. I think it's kind of cool to think, are these platelets that are being made in the oral cavity different than platelets that are being made in circulation? And then, of course, are these oral megs critical in other oral diseases where IL-17 is implicated, or where platelets are also implicated? And maybe taking it a step further, could this finding explain why thrombocytopenic patients don't present with OPC necessarily just disseminated, that in the case of a systemic deficiency in platelets, if there are oral megs still there producing a localized population, a localized source of platelets, does that protect against the fungal infection? All right, and definitely have to thank my group, the current members, the former members. I think the one thing that might stand out is we do what everyone else does, but with a heck of a lot of undergrads, which can be very gratifying and very frustrating at the same time. We have some other projects in the lab. I'd love to talk about them as well at another time. Definitely have to thank Randy Worth and Leah Wuscher for helping us with that platelet depletion model. Craig Morrell has been a lot of help trying to understand, are these really oral megs? Can we believe it? And he's one of the groups that studies the lung megakaryocytes. And then, of course, have to thank the funding bodies. And believe it or not, I did just receive an R15 just this May to support this work. So we're really excited. And I'll take any questions.
Thank you, Heather, for that informative talk, and congratulations on the award. We're going to move now into the Q&A for both speakers. Linqi, if you could come back, there you are. As a reminder, if you could please post any questions for either Heather or Linqi in the Q&A and preface them with who the question is directed to, that would be very helpful. And I will kick off the Q&A with a question to our first speaker, Linqi. Linqi, this is about your very nice cell host micro paper, and is a question from Minju Kim. Dr. Wang, very cool talk is the comment. And then the question is, are the expression/production of SPS and ergothioneine affected by glucose availability of dormant/proliferating cells in the host lung environment?
Okay, thanks for the question. So actually, our lungs are glucose poor organs. So I don't believe glucose play a role in triggering SPS1 production and ergothioneine production during lung infection. And actually, our recent published data supports during Cryptococcus infection, the granuloma starts to form. And during the formation of a granuloma, the immune cells release RS, which stimulates cellular dormancy for cryptococcal neoformans, and which enables the production of high levels of SPS1 and ergothioneine. But actually, we still perform experiments to nail this hypothesis. Actually, so we are very positive for this hypothesis. So this is my answer. Thank you.
Great. So Heather, we have a question from Dr. Mayan Bhatia about basically the translational findings for potentially therapeutic potential based on your observations with megakaryocyte and platelets. Do you want to maybe hypothesize or postulate? Can this pathway be exploited therapeutically?
Yeah, so I think first we have to definitely, find oral, if you're talking about oral megs, right, we have to see if there is a localized population, I think, in humans, right? That's definitely the next step. But I think so, right? So we have some findings in our cell line that I think, right, even if others had connected platelets with Canada and maybe didn't find a connection or found more, if there wasn't a connection, some of the readouts were more vascular in nature. I think what we're seeing is that if we take platelets and we activate them with a fungal component, or activate megakaryocytes, I guess more also with a fungal component, and then the platelets they are going to elicit, right, are those antifungal platelets that may not have the same vascular effect, maybe if it's going to be something bad, that they would be protective in the fungal infection, right, if they were used therapeutically. And then we see that, and then we're looking at, eliciting platelets with TPO, ADP, right? Some vascular agonists versus 17 versus candidolysin, and what some of those functional differences are.
Thank you.
Yeah.
Linqi, we have a question for you next from Julia Kohler. First, Julia, thanks you for this really fantastic talk, and also says that she loves your daughter's art. Yes, that was very nice to share. Julie's interested about treating PF. She says polymyxin, colistin acts by disrupting gram-negative bacteria cell membranes, is not active against gram-positive bacteria. And she has a two-part question. First, what do you think is the target in PF? And then secondarily, since polymyxin, colistin is very toxic in humans, are you looking for other compounds that inhibit that putative target?
Okay, so that's very sweet. Thank you. Okay, so for the first question, my answer is very simple. That is, I don't know. So I don't know the target. So, but my guess is, so for polymyxin B, so we found that when we use 10 to 9 cells, and we use when we increase temperature to enable mutagenesis. Okay, but after 6 times experiments, we cannot identify any mutant resistance to polymyxin B. So that means polymyxin B is a high genetic barrier to resistance for polymyxin B. So this is has a very high genetic barrier to resistance. So for RF new pathogen. Okay, so my guess is so maybe the polymyxin B can bind to the memory lipid. So for if because so if it targets a protein, so that means if there is a mutation in the residues, and the resistance happen. But for the if it targets a memory lipid, so that means the resistant mutant may be the defective in such a memory lipid production. So that will cause a very high fitness cost. Okay, so this is so one possibility. Another possibility is maybe the polymyxin B have different targets. So for RF pathogen, that means even one target has mutation. Okay, so that is not sufficient to trigger a resistance phenotype. So, but we still don't know. So, what is the targets for polymyxin B? So, the second question, I think that is a good question because polymyxin B is toxic. So, but we should take the circumstance into the consideration. So, for RF, it's very easy to develop a pan-resistance mutant. That means we don't have any option to treat this pathogen. So in this case, and you have to find out another option. Although this option is not, the best one. So, but it can treat the pan-resistance mutant, okay, mediated infection. So for patients. So I think so. In this case, the polymyxin B so may be used to treat the patients infected with the pan-resistance mutants. So for this new pathogen, okay, so this is my answer.
Thank you.
Had a question for it's from an anonymous attendee, but they were asking about the number of megakaryocytes in the oral cavity in relation to the amount in the bone marrow. I mean, I guess it's tough to really compare those two spaces, right? Because they're so different, even from a vascular perspective. But and then also you would allude to that there may be differences, or are the megakaryocytes imprinted in the oral mucosa, and do they have different transcripts or proteins being expressed there?
Yeah, so that's actually what we're trying to focus on now, and trying to understand, because if you saw that right there is that population in the sham mice, that even if in any of our knockouts, right, that sham population is still there, right? So we're trying to understand the signals that keep it there, why it's there to begin with, and then also what are some of the properties of those cells as it relates to progenitor properties, right? And are they involved in any sort of trained immunity, and how they're involved in a memory response or anything like that? But yeah, we're trying to understand those cells better for sure. Yeah.
And kind of a follow on onto that, in terms of platelet biogenesis. So I think like Mark Looney's done a lot of intravital microscopy and shown that a lot of megakaryocytes get trapped in the pulmonary vasculature because the capillaries are so small, and essentially they embolize, right? And then they actually generate the platelets right in there in the capillary endothelium. Are your megakaryocytes intravascular, and are they synthesizing
So
platelets?
yeah, we don't see them associated with the vasculature. I mean, we haven't, we definitely need to do some more studies, but yeah, they are not associated with the vasculature. Yeah.
Great. Right, Christina.
Linqi, I had a question about the temperature and mutagenesis of your PF fungal pathogen. I was interested in maybe the kind of broader perspective of whether you think there could be something special about the recent adaptation of this pathogen, if it's because it's a rare pathogen, that it might be kind of different in showing this signature of mutagenesis. Due to temperature, but the stress of being at human body temperature, although it can, grow, is different in this pathogen, or because lots of fungi will have ROS signatures when they're exposed to things in the human host. If this is something we might kind of just generally encounter something specific to this pathogen, or kind of how you think about this more generally.
Oh, this is a good question. Actually, as I mentioned before, and we are testing approximately 800 yeast species for increasing temperature to see so if there is any effect of increasing temperature on mutagenesis. So we found actually this is kind of common. So in some species, and this is my guess. That is, so for some the fungal species. So actually, the 37-degree temperature, this is our body temperature, is not a suitable temperature for them. So maybe we found for this new pathogen, so the endogenous RS generation is from the dysfunctional mitochondria. Okay, so because maybe so the increase in head temperature will cause dysfunctional the mitochondria, and you will see more the intracellular RS generation. Okay, so another thing is so for our the mismatch repairment machinery. So actually, so for their activities, so they need appropriates the temperature. And if you increase the temperature, that means maybe the enzyme activity. So for the machinery does not have the so the proper capability. Okay, so that means the repairment is not very efficient. So to repair the genome mutation. So this is my answer. Okay, Christina.
Thank you.
Thank you very much. That is a very good question.
Thank you.
I had a follow-up question for Dr. Wang. Do you what about the implications of the glucose pathway or the pathway in azole resistance? Is there any, or is it specific to the pathway you outlined today?
So my answer is we haven't tried that. We haven't tried that because for the phenotypic resistance is associated with cyto drugs. So, but for fluconazole drug, that is a fungistatic drug for cryptococcal But I think so maybe some host metabolites have a role so in azole efficacy. I think so maybe there are some host metabolites, but we didn't try that.
Right. Heather, follow-up. So platelets are a large source of PGE2, and of course that's been implicated in Th17 differentiation. So, have you looked at whether platelets regulate, well, the early IL-17 response, but also like a tissue-resident memory Th17 response?
Yeah, we have not yet. But that's a really great question. We've also started to do some, and also to your last question, we've started to do some spatial genomics as well to understand that population and understand the transcriptional profile. So I think that as we get closer to understanding them better, right, we'll see some of those pathways, I think, come out. Yeah. But yeah, we haven't looked.
Okay, great. But did you get TRMs in the model? After the mice recover, you get memory cells?
Yeah, yeah.
Yeah, that'd be interesting to look at. And then the platelet deleter mouse that you have, is it it's conditional? So you could do
Oh
platelet-
yeah, we've done some fun things with depleting platelets through antibody and leaving those oral megs in the oral cavity. Those mice are not susceptible. But also just playing around with the DT. And we're doing that now, just looking in the memory response, right? But you give DT, you pull it back, you can have those platelets come back and go, yeah, so it's kind of cool. Yeah.
Great. Thank you. Christine, should we, I think we've addressed all the questions, right? Any
There's
further?
one last question that just came in. If we want
Okay,
to,
great. Oh, yeah, thanks. Is that for Heather or? Yeah.
is for help. And I think
Okay,
we'll.
also from Dr. Kohler. Okay. Heather, there's a population of healthy humans who are susceptible to oral candidiasis, babies, until they develop adaptive immunity. Have you looked at the role of megakaryocytes in developing, kind of along the same line, adaptive, like platelets regulating adaptive, like CD4 dependent immunity?
Yeah, we are in the middle of it right now. We're about 4 weeks in, so we have to give it a little bit more time. But yeah, that's the next step. Yep.
Yeah, great.
Yeah.
All right. Well, with that, we wrapped a little bit early, but I think it was a really excellent series of seminars and great questions. And we welcome everybody to attend next month's MycoTalks. Thank you all.
Thank you.